Welcome, everyone, to today's presentation, LHON and Gene Therapy. Where are we now? As the board chair of IMP, my name is Kyra Mann. I'm the board chair of IMP and also the CEO of Mito Action. And it is our pleasure to be here today to offer this presentation on our LHON Awareness Day. So we look forward to bringing some incredible speakers to you. Just a few housekeeping items before we get started. This webinar will be recorded and it'll be available on the IMP website in the coming days. If you are Italian speaking, you should see an option on your screen to enter the translation mode where we have a translator who will be translating today's presentations from English to Italian. So only Italian. So if you have any questions or having difficulty accessing that, if you can just drop a note in the chat to any of the hosts or the panelists and we'll get you set up. But we're honored to have that feature available today. So let's get started. So just wanted to give a little bit of overview about IMP and share with you the work that we do. For those who don't know IMP, IMP is an international patient organization that represents, we are the umbrella organization for all patient advocacy groups, international, and IMP has been in existence just over 10 years. And so our mission is to increase the quality of life for people with mitochondrial disease by facilitating cross-border cooperation and collaboration among national patient organizations. We are affiliated with Eurotas, CORD, which is the Canadian Organization for Rare Diseases, as well as the North American National Organization for Rare Diseases in the United States. The objectives for IMP are to share best practices, information, and knowledge in order to promote early diagnosis, develop appropriate care pathways, and to improve the clinical management of mitochondrial disease. And so we have a medical advisory board that helps advise IMP in terms of the clinical research initiatives that we participate in so that we can fulfill those objectives. We also are focused on being an international bridge between patients, clinicians, scientists, industry, and policymakers. So we work closely with clinicians and patients, researchers, and our industry partners globally. And our final objective is to promote and advocate for speedy development of treatments and cures. We currently have 18 member organizations, as you can see, that spanned across the globe, and we always welcome new organizations that are interested in joining IMP with an objective so that IMP can create a mentorship program. So even if you're a really small organization that's just getting started, or you're a larger organization supporting patients, we are here to support one another, to learn from one another, and to help ensure that all of our organizations have the resources that they need to support their patients in their local community. So some of the main initiatives that IMP is focused on, one is the Sympathic Project, and this is an initiative focused on drug repurposing. And so it's a consortium that's looking to identify drugs that are suited for repositioning as interventions in 9 rare disorders. And there is one mitochondrial disease that's part of this project, which is Leigh syndrome. So we're excited to be a part of this initiative. We also are focused on facilitating the review and publication of a list of medicines to be used with caution by Mito patients. This list is continually updated by our medical advisory board, and we have a very active medical committee that is helping to make sure that this list stays updated and current for the patient community. We also are working very diligent on the first global mitochondrial disease registry, our genomic project, and we're hoping that this year we'll be going live with several different countries. And so if you are in a country where there is no patient registry, we would encourage you to reach out to us because we want to make sure that this project truly captures the global patient community and voice. So we're looking forward to this being launched and going live in 2025. And then Mito IMP also provides one-on-one support through partnerships with all of our patient advocacy groups and our member organizations, and specifically Mito Action and the Mito Foundation in Australia are leading that initiative. So any patient, no matter where you are in the globe, if you reach out to IMP, we will have an individual to provide one-on-one support with you, connect you with a local clinician, answer questions, connect you to other families, any type of support that you may need on your journey with mitochondrial disease. And finally, of course, we're in World Mitochondrial Disease Awareness Week, which we're really excited to continue to expand this program and collectively and globally raising awareness for mitochondrial disease. And so today is LHON Awareness Day. Last week, we celebrated TK2D Awareness Day, and some of the other initiatives we'll have on the 21st, Light Up for Mito. So make sure you're getting your green light bulbs and putting them on your front porch and sharing those photos with us. And then also the Faces of Mito campaign, which allows us to share the stories of some of our individuals who are affected by mitochondrial disease in an effort to raise global awareness. Okay, and that's just the overview for IMP. So I am going to turn it over now to Paula Morandi. Paula is a member of the IMP Board of Directors and is also an LHON patient as well. Paula?
Thank you, Kyra. And I'd like to thank Mito for supplying the interpreter service for the Italian patients who are connected today. And for this reason, again, I would like to stress the importance of speaking slowly to all speakers because the interpreter is by herself the 2 hours. I'd like to say just a couple of words about the LHON Awareness Day, which is something I really, really cherish. It was founded by IMP in 2019, and this is the 5th edition. Every year we have chosen a theme. We started in 2019 with the theme of diagnosis and therapies. And on this occasion, we also put together a lovely brochure that we distribute in all the hospitals wherever we can with eye doctors, neurologists, and so on. But it is also on the IMP website in Italian, in English, in Portuguese, and in Spanish. We hope to be able to translate it soon in other languages. Why is it important to celebrate LHON Awareness Day? I have been affected by LHON myself for the last 32 years. And today I still see the difficulty in diagnosing LHON. This is one of the reasons why it's so important to spread awareness, to talk about it, because many eye doctors still don't know when they come across a patient who has no eye pain, who has loss of central vision. They often stop at the eye and omit to look at the back of the eye, which is where they need to look for the ganglion cells and where the damage begins. What happens when you develop LHON? Your life changes drastically, of course, for the worse. And the patient has very little time to even understand what is going on. In many cases, patients don't know that they have a rare disease in their family, as this was my case. So it's a huge surprise. It doesn't only affect the patient, but LHON, like all other mitochondrial diseases, affects the whole family. There are feelings of depression from the very beginning because it is a quick disease. It can take up to only just a few weeks or months to stabilize, and it's just too sudden. Also, there are feelings of guilt because it is passed on from mother to children, and often we find that mothers feel guilty without having any real guilt. It often starts with patients in their school age, and difficulty in suddenly following the teachers, seeing the board, and reading books is all very quick and very, very difficult. We find difficult, therefore, at school and at work. There are many difficulties in relationships. We see young boys and girls wanting to do the things that all teenagers do, but of course not being able to drive, not being able to ride a bicycle, not being able to take a scooter or a motorcycle. All these things make us different from others. There are many, many difficulties that we encounter, like even being believed or being understood, often by even our own families. Many doctors, even today, they still think that the patient sometimes exaggerates and even sometimes a little crazy. Theodore Leber gave his name to different diseases, and we must learn to differentiate among them. LHON, Leber's hereditary optic neuropathy, for example, is very different from Leber's congenital amaurosis. It's very, very important for patients to look for patients' organizations and to be guided by them, by us, in the right direction. I thank you all for being here today, and I'd like to pass the floor back to Kyra.
Thank you, Paula. I appreciate that. So we're going to get started with our first speaker. It is my pleasure to introduce Dr. Magali Tayel, a distinguished expert in ophthalmology and gene therapy. Dr. Tayel completed her doctorate of medicine from the University of Paris in 1993 and went on to earn her associate professor degree in 1998. After over a decade of clinical practice, she held leadership roles at leading pharmaceutical companies, including Lilly and Pfizer, where she made critical contributions to drug development. Now serving as Chief Medical Officer at Gencyte Biologics, she leads groundbreaking clinical research on gene therapies for rare ophthalmic diseases such as Leber's hereditary optic neuropathy and retinitis pigmentosus. Please join me in welcoming Dr. Tayel.
Thank you so much, Kara, for this very nice introduction. And thank you for really inviting us for this day and for this presentation. So thank you to IMP team. Let me just share my screen. Okay. So thanks again. Very happy to be there. Of course, today we're going to focus this presentation on our development in Leber hereditary optic neuropathy. And we are at late stage development for a gene therapy called lenanogenol Parvovec, and the brand name is Lumivoq. And I'm not going to explain the disease. It was so well expressed by Paula Morandi just before. Just as a reminder, as you know, there are several mitochondrial mutations leading to LHON. And the most frequent one is the mutation on the ND4 mitochondrial gene. It represents roughly 75% of LHON patients, and it's also the mitochondrial mutation leading to the most severe clinical form of LHON. So Gencyte Biologics developed a therapy, a gene therapy, to overcome a mitochondrial mutation, and this gene therapy called Lumivoq specifically targets the mutation on the ND4 mitochondrial gene. The product is injected via intravitreal injection, so it's a very easy procedure. It's not invasive, it's not surgery. Why? Because by injecting into the vitreous, it's the best way to target the retinal ganglion cells which are affected in this disease. Today we have an unprecedented data set. We have 4 phase 3 trials. One is still ongoing, and in total, 174 patients were treated unilaterally or bilaterally via intravitreal injection with a drug in the phase 3 trials. We also have early access programs, and in total, 63 patients, ND4 patients, were treated through these early access programs. So in a summary, if you look at the results of these phase 3 trials, so in total 4 phase 3 trials, in one trial called Reflect, half of the patients were bilaterally treated and half were unilaterally treated, so one eye injected and one eye with placebo injection. And for the other phase 3 clinical trials, patients were unilaterally treated, one eye injected with a drug, and one eye receiving sham injection. So to summarize, if you look at the improvement versus nadir, which is the lowest point of vision of each eye of each patient, the treated eyes at 3 years in Reflect, there is an improvement approximately of 20 letters. And for the treated eyes at 5 years for the other clinical trials, it's roughly 22 and 23 letters improvement. So these are the results for the treated eyes. What was unexpected is actually that the sham and placebo eyes, so the untreated eyes, also improved. So in the Reflect study, there was an improvement of placebo eyes of 14 letters at 3 years, and for the other trials, approximately 20 and 18 letters at 5 years post-treatment. So we tried to understand why we observed what we can call this contralateral therapeutic effect, meaning that treated eyes improve, but also untreated eyes. To do so, we performed animal studies in mice and in monkeys, and we could demonstrate actually that when these animals are treated unilaterally with a drug, with Lumivoq, the Lumivoq DNA is transferred from the injected eyes to the contralateral uninjected eyes. And these data have been published. Recently, Professor Valerio Carelli and other people found out from 2 patients unilaterally treated in our clinical trial from the rescue study. So 2 patients unilaterally treated with a drug. These data showed that actually we could found and quantify Lumivoq DNA in the non-injected contralateral eyes of these patients. So in the same way it was demonstrated in animals, we could demonstrate the transfer of Lumivoq DNA from injected eyes to contralateral uninjected eyes in patients in clinical trials. But having said that, when you look at our clinical trials, even if we do understand why now we have this contralateral therapeutic effect, or we have insights why we have this contralateral therapeutic effect, we lost our control groups in the clinical trials because the sham and placebo eyes improved. So to overcome this issue, we decided to compare the data from patients treated in our phase 3 clinical trials to an external control group of ND4 patients who were untreated from natural history studies. So treated versus untreated patients. Of course, in this untreated group, it's coming from publications. These patients were all ND4 in terms of mutation, and also we matched the comparison regarding the follow-up post-vision loss to have a solid comparison of the two groups. And what happened when you look at the evolution of visual acuity of these patients, so the treated patients, you see that the treated patients got an improvement of visual acuity over time, which is continuous, which is persistence over a long-term follow-up. And it's something that we know. Untreated patients got actually no improvement and even a worsening in their visual acuity. And the difference between the evolution of visual acuity is clinically meaningful. We also decided to perform meta-analysis, meaning that you use many studies from different groups. So we identified 3 groups of interest: untreated patients from natural history studies, idebenone patients. As you know, idebenone is a product approved for LHON in Europe. So patients treated with idebenone, and also patients treated with Lumivoq. And for each group, we perform a meta-analysis, meaning we are pulling data from many trials. And to have a meaningful comparison between these 3 groups, we used a common criterion of recovery, which is called the clinically relevant recovery. This criterion of improvement of recovery has been endorsed in a consensus of LHON experts. That's why we use this common criterion for these 3 groups. And the results are striking because there is a gradient of efficacy if you look at the 3 groups. So for untreated patients, the responder rate at patient's level, meaning the rate of recovery, is about 20%. For idebenone treated patients, it's approximately 40%. And for Lumivoq treated patients, it is close to 70%. It means that Lumivoq triples the rate of recovery versus no treatment and doubles the rate of recovery versus idebenone. We have also data, as I told you, in the early access programs. 63 patients were treated, patients coming from Italy, from France, from the US, from UK. And the evolution of visual acuity over time for these patients post-treatment is comparable to what is seen in clinical trials, meaning a meaningful improvement of visual acuity over time. We have right now follow-up over 2 years, and we'll be going on with this follow-up. Safety is also important, as you know. The safety of the drug is really good. There is an excellent systemic tolerance. Why? Because the biodissemination of the product is limited in the blood and limited in the urines and limited also in the tears. So excellent systemic tolerance. The main ocular adverse event is intraocular inflammation, which is classical with gene therapy. It's frequent, but the good thing with Lumivoq is that this intraocular inflammation is mostly mild and is responsive to conventional treatment, mostly corticosteroids eye drops alone. And I'm gonna finish on the regulatory pathway because I think it's extremely important to share with you where we are regarding the pathway to move to approval of this gene therapy. So we're going to restart early access program in France very soon. And importantly, I would like to stress on feedback from FDA, EMA, and MHRA, so UK regulators as well. EMA and FDA asked us to perform a new phase 3 clinical trial to confirm the efficacy of the drug. Why? Because we got, as you know, this contralateral effect, and we had comparison versus an external control group. So they would like to see a confirmatory trial, phase 3 trial, comparing a bilateral treatment with Lumivoq versus a pure control arm, so meaning untreated patients in a randomized clinical trial. And we're gonna launch this trial next year in 2025. And last but not least, the MHRA had for now a different position. So these are the UK regulators. They asked us actually to submit our regulatory file with all our data, with indirect comparison versus natural history, comparison with Idebenon, all the data we gathered, and they asked us to submit this package in 2025. It will be done to assess these data and to see if we're going to move to approval directly with this data package or if we're going to ask for an additional trial. So there might be, I would say, a shortcut or a shorter pathway for approval in the UK. So I would like to warmly thank all the patients and especially the patients participating in this program. Nothing is possible without you. And thanks also to investigators and study teams for participating in our clinical trials. Thank you for your attention. And I'm going to hand over to Kyra.
Thank you so much, Dr. Tayal. We appreciate the insights that you just shared with us. And just a reminder to everyone, if you have questions, please drop them in the Q&A, and we will do a Q&A session at the very end of the presentations. Thank you so much. So we have a little bit of a change in our agenda. Dr. Lee is stuck in the airport, and we're hoping he'll be able to join us towards the end. But next, I would like to introduce Dr. Chiara Lamorgia, and I apologize if I'm mispronouncing your name. It's my pleasure to introduce an esteemed associate professor in the Department of Biomedical and Neuromotor Sciences at Bologna University, Italy. Dr. Lamorgia has made significant contributions to the field, authoring numerous peer-reviewed publications and presenting at national and international conferences. Her work has earned recognition from experts around the world, and she is actively involved in various scientific societies. With a career decorated by prestigious awards and funding, Dr. Lamorgia is a leading voice in biomedical research. Please join me in welcoming Chiara Lamorgia.
Thank you very much for your nice presentation. I'm very, very happy to be with you today. So I will try to tell you about LHON and gene therapy by the clinician perspective. You will see in my presentation some overlap with Magali's, but I will try to give you the clinician point of view. These are my disclosure. So a very brief introduction on the disease. So this disease is due to mitochondrial DNA mutation, and the three most common mutations leading to Leber's hereditary optic neuropathy are the 11778, which is the most common, and then you have 3460 and 14484. In general, these three mutations account for the 90–95% of cases. This disease has some peculiarities. One of these is the incomplete penetrance, which means that having the mutation doesn't mean that you will be affected. It's only a small percentage of patients who are carrying the mutation became affected. There is also a gender imbalance, so males are usually more prone to be affected than females, and the usual peak of age at onset is between 15 and 35 years, even though we have cases very young, even children, and also very late onset LHON cases. There is some possibility of recovery, but usually this recovery is not complete. One of the characteristics of the disease, since it's due to mitochondrial DNA mutation, is that the mutation is transmitted along the maternal line, because only females can transmit the mutation to the siblings. And the specificity of the target of this disease are retinal ganglion cells. So, which is the typical picture of this disease? Usually, I said there is a male prevalence, so you have young adult male, but you can have also children, very old people, and female, of course. Can be present a family history positive along the maternal line, but we have very big families with just one individual affected. And the disease, as Paola mentioned, is typically becomes very acutely, so the patient is referring to the clinician a very acute loss of vision, which is usually very rapid and can be initially unilateral. In 25–50% of cases can be bilateral at onset, but usually the second eye in the unilateral case becomes affected quite soon. The differential diagnosis, which is the most common, is inflammatory optic neuropathies, but is very atypical. I would say never happens for Lebers to have pain with eye movements, even though some of the patients are referring to us some headache or maybe discomfort, but it's not real a pain with the eye movements. And another point is that there is no recovery. There are some specific findings at fundus examination, which are specific for this disease and can help in the early diagnosis. You can see, in fact, the presence of hyperemia of the optic disc, also microangiopathy with vessel tortuosity, and also there is this specific finding, which is the pseudoedema of the retinal fibers around the optic disc. As the disease progresses, this hyperemia resolves and there is an evolution to optic atrophy. Visual fields shows initially a central scotoma, which progresses to a generalized defect over time. And OCT helped a lot clinician in making the diagnosis, because it's showing this typical pattern of pseudoedema, which starts in the temporal side of the optic nerve and then progresses around the optic disc and evolves then to optic atrophy. There is no leakage at fluorangiography, a difference with inflammatory papillitis, and usually brain MRI is negative. So the use of OCT helped a lot our work, because now we can follow the disease course and we can recognize the different stages. So there is an asymptomatic stage, which means carrying the mutation without showing any sign of the disease. And then when the disease starts, there is this subacute phase, which is between the onset and 6 months. Then you have the dynamic phase between 6 and 12 months. And then you enter this chronic phase, which is after 1 year. And each of these stages is characterized by specific OCT findings. At the moment, the only approved therapy for LHON is idebenone. But I will tell you about the option of gene therapy, which at the moment is available only for the 11778 mutation. So as I said, idibenone is the only approved drug for this disease. The approval was in 2015. And for example, in Italy, we can prescribe idibenone according to the rules of rare diseases using an IFA web prescription in patients older than 14 years old and with disease duration less than 1 year from the second eye. The treatment is at 900 milligrams per day and can be continued for 2 years until a plateau is reached. Moving to gene therapy, I can be fast in this slide because Magali already told you about the design of this therapy, but in brief, you have this intravitreal injection using AAV2 vector. Inside this vector, you put a construct where ND4, which is the subunit which is affected in this mutation, is recoded and then imported to mitochondria. Different phase 3 clinical studies, as Magali mentioned, has been conducted. The first two were two trials where you had one unilateral injection, so you have one eye sham and the other was injected. The difference between these two trials is that for the rescue trial, the patients treated were with disease duration between 0 and 6 months, for the reverse trial between 6 and 12 months. The other trial is the REFLECT, in which the patients received either bilateral injection and another group received in one eye the injection and in the other eye placebo injection. So in the second trial, you have a group of patients who received bilateral injection. Moving to the results that already were shown by Magali, I just want to emphasize, as you can see from this graph, that as the disease progresses, you have this worsening of the visual acuity, which is continuing despite the treatment, but then you have this recovery of vision. And if you compare the two lines which are related to the reverse trial to the lines referring to the rescue group of patients, you can see a difference in the sense that maybe there is a better improvement for the patients treated a little bit later. And this is something that is still not very well understood. Then, and this is something that already Magali showed, if you compare the results of people treated with gene therapy to the untreated population, you can see this difference, which is quite significant in the treated group, pulling together all the results to the untreated group. And for the bilateral injection, as you can see, there is a better outcome, visual outcome, for people receiving the treatment in both eyes. As already said, after the conclusion of the phase 3 clinical trials, 63 patients were treated thanks to this expanded access program, which was provided in patients coming from the US, France, Italy, and UK. And as you can see from this table, 67% of patients received bilateral injection, and the mean disease duration for people treated through this expanded access program was 11.3 months. And you can see this improvement over time, and this was these are data obtained from a 2-year follow-up. And overall, the improvement from the nadir, which is the worst visual acuity achieved for each patient, was very similar to the results already published for the bilateral trial, the REFLECT trial. However, there are still, this is something that we are still debating. There are some unsolved unanswered question, I would say, in these gene therapy results. One of these, and Professor Carelli will talk a lot about this in the next talk, why there is this bilateral recovery of visual acuity in the unilateral injected eyes. Another question that I already pointed out, it seems that patients treated after 6 months in the dynamic chronic phase of the disease are experiencing some better outcome than patients treated earlier. Why is that? And also, as I already mentioned, it seems that this therapy, as this applies also for Idibenon, this gene therapy is not able to prevent visual loss at least in the acute stage of the disease. And other two open questions are, which is the efficiency of retinal ganglion cells transfection, and is the imported ND4 efficiently assembled and integrated in complex 1? And with this, I'm finished, and I would like to thank you for the attention, and I would like to thank you a lot, the people working with me in Bologna and all the collaborators. Thank you very much.
Thank you so much, Dr. Lamorgia. We appreciate that. When a few questions came in, so we will get to those at the end of our presentations. So thank you so much.
You're welcome.
Okay, up next, we have Professor Valerio Carelli. I'm honored to introduce Professor Carelli, a distinguished neurologist at IRCCS Istituto delle Scienze Neurologiche di Bologna and the University of Bologna, Italy. Professor Carelli is a leading expert in neurodegenerative diseases and mitochondrial disorders with an extensive career dedicated to groundbreaking research and clinical advancements. His work has had a profound impact on the understanding and treatment of neurological conditions, earning him recognition as a global leader in the field. Please join me in welcoming Professor Carelli.
Thank you very much for the introduction, and I apologize for my voice, but I will try to be as clear as possible. I hope you see the slides. I will expand a little bit on what Magali said about the bilateral recovery of treated patients. I have disclosures. I essentially participate to all the clinical trials involving mitochondrial diseases, but I don't own any share of those pharmaceutical companies. I just act as a consultant or as a PI in the clinical trial. This is the third time that you see these slides. I apologize for that, but here the point is that there are a couple of things, 2 or 3 things that has been already pointed out by Chiara, which open some questions. Any drug we give to Leber's patients, either Idibenon or gene therapy, patients deteriorate the visual acuity over the time, following pretty much what is the natural course of the disease. And then these therapies are more or less efficient depending on the approach. We just saw from Magali that the gene therapy seems to be superior to Idibenon in recovering vision in the following period. And surprisingly, in gene therapy patients treated after 6 months, the visual outcome seems to be better than those treated earlier, which is completely counterintuitive. The idea about the bilateral recovery, despite only one eye was injected, has been already introduced by Magali, and a few studies in animals showed that there is the possibility for the diffusion of the viral vector from the eye which is injected to the contralateral eye. This is a cartoon showing that actually the virus in the animals, we're talking about primates in this case, can be found all along the visual pathway from the eye in the brain to the and through the visual pathway all the way down to the visual cortex, but can be found also only in the anterior part of the visual pathway of the eye, which was not injected. So the additional information I will be providing in this talk now is concerning about the case of two patients which unfortunately died for unrelated causes, not related to the gene therapy, of course, but we are very grateful to the families which approved the possibility to do the autopsy and to study the eyes. So this is a study of post-mortem tissues of two patients which have been unilaterally injected with the gene therapy. And I only want to point out a difference between the patient 1 and patient 2, because patient 1 had a clinical history which was very severe, and this is reflected in what we found in the eyes and the optic nerves. So the patient had a very severe optic atrophy with a very dramatic loss of retinal ganglion cells, while patient 2 was a much milder case with a good preservation of retinal ganglion cells and optic nerve axons, which were essentially lost only on the temporal side, which typically is the papillomacular bundle, which is the most vulnerable part of the nerve that degenerates. So this is a fundamental difference between the two cases, which might help to understand a little bit how the virus may actually diffuse from one eye to the other. The techniques that we used for studying the tissues recovered from these two patients were actually the classical histopathological assessment with different kind of stainings. And then we used something called laser capturing. So through a laser, we can actually choose to pick up specific pieces of the tissue with the specific cell types and layers, and then extract the DNA from there and assess. In this case, we were most interested in assessing the presence or the absence of the viral vector, which is the therapeutic agent of the gene therapy, through a technique called digital droplet PCR. So I'm not going into the details of the description of each of these histological sections, but what we can say is that patient number 1 had a clear-cut evidence of some chronic inflammatory reaction in the optic nerve head, which is something that has been seen in patients, and not all of them they have it, and the severity is very different, and is usually resolved by an appropriate anti-inflammatory steroid therapy. So this is just to say that in this case, we had evidence in the injected eye of some inflammation. Contrary to the patient 1, patient 2 did not have any evidence of inflammation, and the only thing that was remarkably different from patient 1 was the abundance of retinal ganglion cells, which were very depleted in patient 1, and as I said, are still present in a conspicuous, in a good number in patient 2. So getting to the key data, the key results, here you see that in the injected patient, in the injected eye of patient 1, the amount of virus, as expected, of the viral vector, the therapeutic agent, as expected, was very high. And a little bit of the vector was also found in the non-injected eye. So this is a proof of principle that the therapeutic agent somehow can diffuse from the injected eye to the uninjected eye. Interestingly enough, the same thing applies to the second patient, And if you look in the details of the structures that were analyzed, so here is the entire retina analyzed, then we have been able to dissect only the vessels or only the photoreceptors and the choroid or only the optic nerve. And if you compare in the patient 2, the injected eye and the non-injected eye, you can see that you find a fairly good amount of the viral vector in the different structures. Overall, what we think is that having more axons, the amount of the viral vector that has been able to be transferred from one eye to the other was superior in patient 2 as compared to patient 1. Another information which is compatible with the way we do the injections in the eye is that most of the vector was found in the area of the retina which is targeted by the therapy, which is around the macula and not on the nasal side. So this is the part of the retina that we want to rescue in order to save or rescue the visual acuity in the patients. I will skip this other graph and get to the very last and preliminary data. This has been done the last 3 months. I want to be very cautious in interpreting the data because these numbers are absolute numbers not corrected for the surface area of the specimen that we picked with the laser capturing. So please don't look at the amount because this needs to be corrected for the surface. However, what we see here, and this is different from the previous data which were referred to the retina, here we have been able to cut at different levels the optic nerve itself. And you can see that in both nerves, in both patients, you see a good amount of the viral vector, which seems to be more abundant in the temporal fibers of the patient too, because he had more axons. As I said, this case had spared retinal ganglion cells and axons. And the other important point is that we recognize the viral vector in the meninges. And this is something that we still think about because if we privilege the hypothesis that the viral vector diffuses through the axons, the meninges means being infected by the viral vector means that also other routes could be involved in the transfer of the viral vector from one eye to the other one. Last but not least, also the central artery, so also through the vasculature, the viral vector could be traveling around. I will leave you with a number of some conclusions and a number of open questions because this is an evolving story which provides interpretation of what we observe with the clinical trials. We have a clear-cut evidence from the post-mortem studies in these 2 patients that in humans, if you inject one eye with any kind of gene therapy, probably you will end up to have some of the therapeutic agent being transferred on the other eye. This is very important for the design of the clinical trials in the future because the idea that seemed to us very smart at the beginning of non-injecting one eye and keeping that eye as a control is not valid anymore. We also have the evidence that the viral vector is transfecting efficiently the retina. And of course, there is a difference, and this poses the problem of how we value this transfection. There is a difference between the injected eye and the other eye. And the question is the amount of viral vector of the therapeutic agent that diffuses to the other eye enough to justify the bilateral recovery? Consider that the injected eye is always superior to the non-injected eye in the magnitude of the recovery. And one idea is that the route followed by the viral vector to go from one eye to the other are the axons, but we are not excluding other possibilities. As we see, we are getting some unexpected results. Meninges carry the viral vector and the blood vessels as well. And finally, in order to understand how efficient this therapeutic approach is, we managed to have constructed cell lines without the ND4 subunit. So we don't have the competition between the ND4, which is provided by the therapeutic agent, and the mutant ND4, which is provided by the endogenous mitochondria of the patient. And we are confident that studying and revisiting in modern times all the steps of how the viral vector produces the therapeutic effect will be rediscovered through these studies which are upcoming. And I think I finished with this. I thank my team in Bologna and a huge number of colleagues that over the last 30 years we have been working with on Leber's hereditary optic neuropathy all over the world. Thank you very much for listening.
Thank you so much, Professor Corelli. We appreciate all of the hard work and your tireless efforts on behalf of this community. Thank you so much. So next we will have joining us Julian Seychron, who is the president of the LHON French Association and also a person living with LHON. Julian, welcome.
Hi, everybody. I'm Julian. I'm from France. And just thank you for the invitation. I'm here as a witness because I have been detected from LHON 10 years ago. It was in October 2014 when I was 37. It was very brutal, as Paola said, and then I lost my view, the sight, vision, and color in less than 3 months. So at December 2014, I lost everything. It was very quickly. I just asked to the doctor. I have the opportunity to meet Dr. Vignal, who is a specialist of LHON in France. I asked the question, like everybody asked the question, okay, what is the remedy? And she told me there is no remedy, only Roxanne. Okay, so I took Roxanne. He said no effect on me. I say, okay, so life is going to be very, very difficult. And then 3 months after, so it was 6 months after on 2014, it was in April 2015, I have a call from Dr. Vignal, and she told me, I have one last place on the protocol with a gene therapy. I say, okay. Do you want to come in this? I say, okay, I can see 0 letter on ophthalmologic board, 0, 0. I don't recognize color, I don't have sight, so let's go. I say, you have to make understand, okay. I say, let's go. And so I was the number 15 human in the world, so it was 10 years ago, just to be injected. And I think that doctor, nobody knows where they were going because it was the beginning of the gene therapy. And so I was on the hospital, the 15 in France, in Paris. It was my second home because I have to do some check of the blood, of the urines, etc., on the ophthalmologic board. And the ophthalmologic board was white after 1 month, 2 months, 3 months. I say, okay, let's go on holidays. And after holidays, it was 4 months later, the injection. I was injected by one eye because they didn't know where they were going 10 years ago. So they injected one eye. And now they are injected 2 eye, both eye. And then 4 months later, I was on the ophthalmologic board on the hospital. I say Z2E. Okay, I say, okay, I can see, I can read. 3 letter, okay, well, why not? And so day after day, week after week, was better, you know, the sight. And I was working in the streets and say the traffic light. I say, oh, what happens? It's red, it's green, it's great. And I think that people were around me say he's crazy. He's staying on the traffic light, and I say, oh come on, Julian, it's color. And then after a week, I was in the hospital and checked, and on the magic board, I can see now on the eye injected 15 letter 6 months after, and on the non-injected eye, I can read the letter, etc., and I can see my face. Okay, I can see my face on 20 centimeters. Sorry, I can see that my beard is becoming more than white than green than brown, and after I can see already what I can eat. I say in my plate, you know, it's fish, it's vegetable, it's not gray. And what I was drinking some wine, I say, okay, it's some white wine, it's some red wine, and you know, week after week, month after month, because you lost everything, sight, view, and color in 3 months, but the improvement and the recovery with the injection in one eye is very slowly, day after day, week after week, month after month, and at least 1 year and a half, it was the top of improvement for me. And now I can read on the ophthalmologic board more than 35 letters on the left eye, which has been injected, and about 20 letters on the eye which has not been injected. And also doctor talk about the collateral effects right now. It was 10 years ago, and the first question I ask when I get with this recovery to the doctor is say, did I agree to all I want, whether I get it in 10 years, in 5 years, in 10 years, and say, we don't know. But I can tell you as a witness that 10 years ago I was injected. 8 years ago it was the top improve of my recovery. And now I didn't lose something. And I set up an association in France for all the guy who have been injected on EAP because it was in 2020 in France and the AP, and it's wonderful that Magali told us that EAP will continue and as soon as possible in France, in Italy, etc. And so in my situation, there is only a patient who has been injected in France on one eye or two eyes, and just to coach them to say, okay, you're not going to be in the black, you're going to lost everything, but you're going to be injected, it's going to be very slowly, but you're going to recover some color, some size, some view, etc. And which is incredible, and it's more than 80 patients in my association have recovered sight, some view, and color. And so it's a change in the life. Okay, you are not going, not able to drive, but now I can ski, you know, I can read some newspaper, big title, okay, and after I have Zoom, etc. I can recognize my face, even if I have to be close, okay, to the mirror, but it's a change of life, you know. When you're walking the street, etc. We are in the subway in Paris. You can read the station. You're not lose. So okay, you have to get some voiceover, some help, some Zoom text in the laptop, etc. But it's so a big change. So I hope that some study and some EMA, FDA, MHRA going to understand that patients have to have this gene therapy very quickly, because after 2 or 3 years, I don't know exactly the timing, they cannot be injected. So it's as a witness, I think that there is some question after. That's why I want to say, it changed your life. it was 10 years ago. It was the beginning of the gene therapy. It works. I'm happy it works. I'm happy for all people that have been injected, and I would like to have more people in the world to be injected, because we know it works. And for people who can read, I just have a book, I wrote a book on that. I'm sorry, it's just in French. I try to have some translation, but some money, so I'm going to try to make the translation for new patients. And what Paula said is very important, that it's not only patient, but it's from the husband, the people around patients. It's very difficult to make understand what we see, and they say, Julian, now you can see what. Let us say, I can see color, I can see what I'm eating, I can see maybe I can see my wife, I can see the lipstick of my wife, if something changed, okay, I cannot drive, and so what. So that's why I think I got only 10 minutes. I don't have slide, I'm sorry, just on vocal. And afterward, if you have some question, do not hesitate. I think just have some 10 minutes. So
Thank you so much, Julian, for sharing your perspective and your experience with the trial. We'll have an opportunity for people to ask questions as soon as we're finished with the final speaker. So stay tuned with us, okay?
okay.
So up next, it's my pleasure to introduce Caroline Pothet, the head of advanced therapies and hematological diseases at the European Medicines Agency, EMA. With over 20 years of experience in pharmaceutical industry, including a decade in oncology and 7 years in immunoinflammation drug development, Caroline brings a wealth of expertise to her role. She also serves as the regulatory science committee of the International Rare Diseases Research Consortium. Prior to joining EMA in 2020, she held key positions in regulatory policy affairs and global R&D. Please join me in welcoming Caroline.
Thank you. Can you see the slide as a slideshow, just to check?
We can, yes.
Okay, perfect. Thank you. Thanks very much for the invitation and for the nice introduction. So indeed, I'm working for the European Medicine Agency. And as a disclaimer, what I'm going to present is really an overview of the role of the agency in the development of gene therapies. However, I'm not directly involved in the assessment. So it's really more of a personal presentation that you would get from me. So who we are, the agency, actually, it's not just a European Medicine Agency that's based in Amsterdam, but the European Regulatory Network is composed of approximately 4,000 experts across Europe. And we have 7 scientific committees. And the important ones for gene therapies would be the Committee of Advanced Therapies, which I will mention, to which the CHMP, the Committee of Human Medicinal Products, delegate the assessment of ATMPs, and then the PRAC, which is a Pharmacovigilance Review Committee for Safety Information. And then we also have amongst that a management board and involvement from a number of stakeholders, and that include European Commission, civil society representatives, so patients and healthcare professionals. So what we do is we support the development of medicines, and that's both human and veterinary. We are responsible for the coordination of application for marketing authorizations, and those are submitted to the EMA, which we call the centralized procedure. And gene therapies, which are ATMPs, then are mandated to go through the centralized procedure. We have also the mandate to monitor the safety of those medicines across their life cycle. And then we do provide a lot of information and are reviewing any patient's summary of product characteristics. something that we do take very seriously, the conflict of interest. So we do have a policy which is currently undergoing a review, because we actually look at it on a regular basis based on experience. Everything that's assessed by the committees is peer review. So we have already in the European system a rapporteur and a co-rapporteur. So these are the lead member states who will perform the review. But also there is peer review across the system, because all member states who are part of the committee actually have the opportunity to raise comments. And a very important point also for us is real transparency. And if you look for information on products, whether they are registered, not registered, then we do publish assessment report. And that's included also for products that are withdrawn. The ATMPs, or advanced therapies, from which is a subcategory of gene therapy, is actually is really managed as medicinal products, but they are special medicinal products. And there was a regulatory framework that was developed around 2007 and established 2 years later, that actually introduced some definition of what should be considered advanced therapies. And with that, we have a mechanism for classification. It did establish a new committee for advanced therapy. We call it the CAT. It made mandatory the centralized procedure, so that procedure that's managed by the European Medicine Agency rather by national competent authorities. It's also reminded that ATMPs really have to be registered. They need to be a clear understanding and demonstration of quality, efficacy, safety, and that include a positive benefit risk. So how the drug works and whether actually the positive effects are above the risk associated with this medicine. It also introduced specific principles around ATMPs, which is a possibility to ask for more post-marketing authorization follow-up of efficacy and safety data. And one of the reasons, and we heard today, is, for instance, gene therapies often are expected to have a very long effect, and it's or even a lifelong effect. And therefore, it's important that patients are followed so we understand the consequences, and that may be different from a typical drug where you take, and then if something doesn't go well, then you can stop the treatment. You can't do that with most gene therapies. And then we also have a certification for small and medium enterprise, by which we are able to review prior to submission their quality and non-clinical data, so the animal data, before they come for submission. So I explained there is actually 3 categories of ATMPs that would follow this legislative framework. So these are gene therapies and are usually linked to genetic diseases, because that's the idea, is really to regulate, repair, add or delete a genetic sequence. And then you also have 2 other types. I'm not going to develop on these ones, but they are called somatic cell and tissue engineered product. And again, all of these products are regulated as medicinal products and have to follow the centralized procedure. So the entry routes for the ATMPs is therefore the centralized procedure. Before they come for submission, they are dealt with in clinical trials, and the clinical trials are actually reviewed at national level. Then there is a scientific evaluation, which is centralized. And as I mentioned, the Committee on Advanced Therapy is the committee that's responsible for the review and evaluation of the gene therapies. But importantly, they are also involved in providing scientific advice when developers are coming to ask advice. And then the decision maker is a committee for human medicinal products who will issue a positive or a negative opinion based on the recommendation from the CAT. And then eventually that gets registered if all goes well, and then it goes to the HTA who will decide on reimbursement and pricing. The CAT is composed of representatives from all member states, but in addition, there are a couple of what we call double members, so they are both on the CHMP and the CAT. And there are also, which is atypical for some of the committees, they also have representatives from clinicians of healthcare professionals who are nominated by the Commission, and also 2 members from patients' organization. So that's an overview of approved ATMP so far. There have been 26 registered, but only 19 currently actively approved, because 7 of them that had been approved previously have been withdrawn. And as you can see, the vast majority of these products are in the middle, so the blue part, and they are gene therapies. I did refer to the importance of demonstration for quality non-clinical, so safety and efficacy data. And these really are critical aspects to be addressed quite early on. And what we've seen especially for ATMPs and gene therapy is that the importance of quality is actually even more critical than for non-ATMPs, because the quality and reproducibility and the comparability of the different products that are manufactured will actually drive the treatment effect and whether this can be maintained. And therefore, what we've seen in many requests for scientific advice, but also during evaluation, that often you see a bit of a similar extent, a number of objection or questions on quality and clinical, and that's not necessarily the case with other non-ATMP products. Now we acknowledge that it can be even more challenging to develop ATMPs or gene therapies because of the complexity and the nature of product, of the type of product, but also the nature of the condition they are treating. And we heard today a very good example. And so the agency offers a number of tools at different stage of development, but the most important one is scientific advice or protocol assistance for orphan disease, where actually the applicants have the ability to discuss with the authorities their plans for their clinical trials or their plan for in terms of the manufacture on the product. And we heard at the beginning of this session that actually a company makes use of that. So to really summarize is, we do, as an agency, our aim is really to get products of good quality and that have demonstrated benefit risk, positive demonstrated risk to the patients. And that can be very complicated to navigate through the regulatory requirements. And therefore, we really make the utmost to ensure that the applicants have the ability to use our support tools, and we are really receptive to providing advice. And as I mentioned, the Committee on Advanced Therapies in this case is involved in as a peer reviewer for all scientific advice requests, and it's very important that we do have this engagement early and as a development move forward. And the other thing that's not on the slide, but we do also have mechanisms to engage with patients' organization and patients throughout these different steps. And that's also something that is paramount to our role in public health. And with that, I would like to thank you for your attention. And I've put a couple of links if you are interested to find more information about the agency. Thank you.
Thank you so much, Caroline. We appreciate. So that brings us to the end of our speakers. So now we will open up for questions. And so I'll start with a few that have come in the chat, if that's okay. So one of the questions, and I'll leave it up to the speakers to jump in and who you think is best to answer the question. The first question is, the trial results are amazing to read. So maybe we go back to our first speaker on this question. Was there any ongoing or long-term impact to the eye inflammation issues?
Well, thank you for the question. So right now we have long-term follow-up, 5 years follow-up for our first phase 3 trials, and 4 years follow-up for the REFLECT trial, where half of the patients were bilaterally treated. We'll get the 5 years follow-up this year, actually, at the end of the year. So globally, what we can see on these results is that the safety is still very good. And what was pointed out by Mrs. Pote is very important regarding safety of gene therapies. So, but still, it's okay, no issue, no new adverse events. And we do know very well regarding the intraocular inflammation, how it occurs and how to manage it. And regarding efficacy, which is also extremely important, the effect is maintained over time for 5 years post-treatment. Of course, we cannot know if it could be maintained for life because we don't have enough follow-up, but from what we are seeing right now, it means that efficacy and safety are okay regarding long-term follow-up.
Wonderful. Thank you so much for that answer. Our next question, someone is writing, my son has been diagnosed with LHON 3 months ago. He's 18 years old and takes idabennin. My question is, what is the process to be nominated or to participate in the clinical trials taking place in Europe or in the United States?
Magali?
Yes, sure. I think it's a great question. So, as pointed out by Professor Caredi, so right now we have a better knowledge of the best, I would say, therapeutic window. Today, the inclusion criteria for the new trial, it's a vision loss between 6 months and 18 months. So, of course, sometimes it's a bit tough because some patients might be, after 18 months of vision loss, and I do understand that. We also have strong data to support treatment within 2 years for early access programs. But again, it's still discussions with regulators. But just to be brief on the response for our clinical trial, you need to have a vision loss of maximum would be 18 months post vision loss to be eligible.
Thank you so much.
So in the specific case of that patient, there is time.
There's time. Wonderful. Thank you for that. This next question is for Professor Corelli. It says, my son is affected for 8 years. His LHON mutation is 3460. Can we expect the investigation to move to his mutation and others affected for more than 1 year?
Yeah, well, I don't think is in the program of Magali's company, but there are other companies which are working on that. I think these are Chinese companies. I have not been in touch with those, so I don't have fresh information. But for sure, there is preclinical work and now possibly also a clinical trial taking care of the other more rare mutations, because obviously 11778 is the most frequent and most important, but there are plenty of other mutations that needs to be taken care of. And actually, this is my question to the EMA representative later on.
Yes, and I'm going to ask, why don't we go ahead and just move into that question, Dr. Corelli? So Dr. Corelli's question for Caroline is, are we going to need for each single mutation all the complicated pathway to approval, or will there be some kind of change or paradigm?
Thank you for this question. We do get it regularly, I guess, through different kind of events or conferences. So I guess what exists already is what we call prior knowledge. So whether it's in quality or whether it's non-clinical data, there is already the possibility to use prior knowledge. And we put together, I think in collaboration with the Food and Drug Administration in the US, for instance, the Prime Quality Toolbox, and that's referred. So that would mean that if a construct, would be the same, and then you're only changing the gene you're going to repair and things, then you would be able to use the prior knowledge. We also, as part of the new pharma legislation, you may have seen that the European Commission has introduced the concept of platform technologies and platform MA. And the idea there, as we understand it, is really to also, in the future, when there will be more advanced, to facilitate this type of development potentially. So I would say at the moment it's on the scientific way, it's there, but it's true that it's not necessarily easy to put in place. But that's something we will welcome applicants to discuss when they come for advice. And then in the future, this might even be more facilitated.
Thank
Good.
you.
Thank you.
Go ahead, Dr. Crowley. Did you have another question?
No. I'm pleased by the answer. Thank you.
Wonderful. Thank you so much. Okay, so the next question, actually, for Chiara, I'm gonna ask a question of you. You talked about expanded access. Can you explain to those listening how expanded access works and how that allows a patient to be able to get access to a therapy?
Oh, yeah. So I was referring to the early access program that Magali was mentioning in her presentation. So I was referring to the 63 patients already treated in the context of the compassionate use, which at the moment is not going on anymore. But Magali showed that this program will start again, at least for France. The situation for Italy is a little bit different because we just submitted, actually in July, we submitted a request to our drug agency, Italian agency, for a 648 application, which is a kind of different situation from the early access program. So the situation is still to be understood in Italy, at least.
I think it's based on national regulations, mainly.
Yeah.
Yeah, definitely. Thank you for that answer. I have a few questions for Dr. Tayel. What is the response to gene therapy equal in every patient? And are there existing markers to predict the response to therapy?
Thank you for this question. Very, very good question. So, if you look at the responder rates, roughly 70% of the patients respond to treatment. And today we don't have any markers or explanations why some patients will respond and others not. We made some progress in terms of timing of treatment, I think. It's not worth, rushing to treat the patients. Initially, we thought it would be better because you have more cells preserved. But at the end of the day, if you look at the results between 6 months and 18 months, it's really good. But again, the response is usually delayed. It's more after 6 months, even 1 year. And 30% don't really respond to treatment. And potentially because, it's a rare disease, we don't have many patients. It's very difficult to find out some kind of predictive factors for response. So sorry, today we don't really know why some patients won't respond.
Thank you for that answer. So the next question is for Dr. Crowley and Dr. Lamorgia. The question is, my son has the mutation 13042GA MTND5 with 11, and he's 11 years old. And he's saying that they read that you investigated this mutation years ago. Is there any information you can provide about that?
Well, this is one of a group of mutations in ND5, which are very complicated. Different from Leber's mutation, the classical ones, these mutations are heteroplasmic, which means they have a certain, these patients, they have a certain amount of the mitochondrial DNA molecules, which are normal, wild type. And depending on the level of the heteroplasm, if you have about 60–70% of the mutant mtDNA, mitochondrial DNA, you may have Leber's. If you have 80%, you may have another disease, which is more complex, like Milas. And if you have even more than 90%, you may have Lee syndrome, which is a dramatic infantile encephalopathy. The situation for this specific mutation is very complex from the clinical point of view, and also for the treatment, we don't have a program for gene therapy similar to what has been done for Leber's. However, because is heteroplasmic, there is a different program which is run by Carlos Moraes in Miami, University of Miami, and Michal Minchuk in Cambridge. They set up editors, mitochondrial DNA editors. They can cut and destroy the mutant and facilitate the expansion of the wild type molecules, and that would be the therapy. But this is still not yet available for clinical trial, but the preclinical level is very advanced. So we hope that in the next future it will be available.
Yeah, he wrote that the mutation is 65% heteroplasmic. So
that's exactly what I said. This is low. You just get Leber's. If it gets high, you get more complicated diseases.
Mhm. Thank you for that answer. The next question is, my son was diagnosed at the age of 9 with atypical Leigh syndrome with the 11778 mutation, typical to LHON. Now age 11, his symptoms are from Leigh syndrome, not LHON. He takes Mito cocktail, which includes CoQ10, and his optic nerve is so far normal. Could the gene therapy presented here or its development help him maintain his vision and improve his Leigh syndrome?
Maybe.
Okay.
Yeah, there is no evidence that gene therapy can be used for carriers. I mean, in this case, he has no affection of the eye, so there is no indication to treat with gene therapy a person without any visual symptom.
Yeah, again, this is a complex situation. First of all, I would suggest this patient to run a supplementary genetic investigation. We recently published that when you have 11778 and you get Lee syndrome instead of Leber's, usually you have the coexistence of another heterozygote mutation in the nuclear genes which encode for complex 1 subunits. So first of all, I would clarify the genetic landscape of this specific case, because there must be something more than just 11778.
Mhm.
Second thing, coenzyme Q doesn't cross the blood-brain barrier, so idibenone would be better than coenzyme Q in this specific case. However, we just show that idibenone is regulated by the polymorphic variants of another enzyme, NQO1. If you have NQO1 variants, you don't get idibenone because it doesn't work. And so he needs to consult with some of us, some of the specialists, and we can give some direction. And lastly, I don't think we are authorized to do, as Chiara was pointing out, a preventive gene therapy for the optic nerve, because this is something that has not been even experimented yet. So you cannot do it. And vice versa, the injection in the eye not necessarily means that you will have any benefit for the brain, because you don't get at that target with the viral vector.
Yeah, because the design of the intravitreal injection is selectively targeting the retinal ganglion cells, right?
Thank you for that. The next question is for Dr. Pothet. How do you assess the quality of a product? the EMA? How does the EMA assess the quality?
Well, and first, I'm not a quality expert actually. I'm a pharmacist by training, but how this is assessed at the agency, it's actually the quality part, and that's true for non-ATMPs and ATMP, is delegated to the biological working party. These are quality experts, and what they look into is really. So first, there are guidelines how to develop a drug that takes from, really confirming what your product is. So you have to characterize your products. You have to run regular manufacturing batches to see that you can reproduce the same thing. And then you look at specifications. So you define some criteria, I guess, quality criteria, which then whenever you generate a new batch will have to meet this criteria. And you also look at stability. So, you have an expiry date on paracetamol tablets. Well, that's based on study. We look whether, the applicant will look whether or not the medicine remains stable. So all of these different elements are put in the dossier provided by the applicant throughout the development, and that's what is being looked at by the quality assessors when the dossier comes to the agency.
Thank you for that. And we'll stay with you for one more question. On one of the last slides in your presentation, you mentioned gene therapy and cell therapy. Can you explain what cell therapy is and how that's different from gene therapy? And are there studies done on that type of treatment?
Yes, so the gene therapies, as I mentioned, is usually linked to the fact you're going to do something to a gene. So it like add a new sequence or repair or delete, and actually it could be cell-based. So it could be based on, you take cells from a donor or from a patient, but then the classification of gene therapy is because of what the intended effect is. So how it's acting on the disease. Cell-based therapies are based on cells that are also from a donor or from a patient, and they are modified. But the effect is not going to be at a genetic level. And also, but one important point as well is these cells are modified from what we get. So it's different from a transplant, for instance. You take something and you pretty much give the same thing. Here you're modifying something which will then have an effect. So I hope that clarifies a bit.
That does. Thank you so much. We've had several people ask questions about potential or existing clinical trials with the 3460 mutation.
So based on what I know, but I have no feedback because we are not, as Valerio mentioned, with the NeuroOft company. There was a trial, at least expected on 3460 from this company. They were expected to participate to this meeting, if I understood correctly, but I don't know much more than this. I have no more.
We have no information.
No more recent update on this.
Thank you so much. And as we learn, if that information does come up, we will be sure to share that with the community on the IMP website as well.
Excuse me, sorry, Kyra. I'm just going to interject just for a second, just to let you know, you're absolutely right. They were supposed to present at this webinar. Unfortunately, the gentleman who was supposed to be joining us has been caught at an airport or on a plane at the moment. I have asked him, or I've asked for them to record the presentation that they were going to give at this webinar. And as soon as I get it, then I will share it with the recording of this webinar as well. So you will be able to get that detail following the webinar. So apologies again that he wasn't able to make it, but I'm assuming that he's up in the air at the moment. So you will get that information in the future.
Okay, thank you.
Good.
Thank you, Joe, for that. Dr. Crowley, I'm going to ask you to help me with this next question. It came through in Italian. We've translated it. So I'll
Yeah.
ask it in English. And then if you wouldn't mind giving the answer in Italian, that would, or re-asking the question in Italian, and then we can move forward and do the answer that way. So the question in English is, I had gene therapy on April 21st, 2021, but after all this time, there's been no improvement. But rather, I perceive a worsening of the situation, the better and the blurrier. Who doesn't have positive results with gene therapy, and what can be done? One hears lately about therapy from China. It's devastating to know that nothing more can be done because unfortunately, one has not had an improvement with gene therapy.
You want to answer?
Will I answer in English or in Italian? I don't know.
But if you wouldn't mind repeating the question, even if you just summarize it in Italian and then answering it in English and then in Italian, that would be great.
So I can summarize in
Yeah,
Italian.
okay.
So la paziente chiedeva, avendo ricevuto terapia nel 2021, non ha osservato nessun beneficio clinico dopo questo tempo, chiedeva se ci sono, anzi lei sperimenta un peggioramento della qualità visiva, chiedeva se ci sono delle altre opzioni e chiedeva anche se si riferiva al trial del gruppo cinese, se ci fossero altre opzioni terapeutiche per coloro che non hanno ricevuto beneficio dalla terapia genica. Questo è in italiano.
So in English, besides idibenone, which is an approved drug in Europe, at least in some countries, and gene therapy, which has been provided in the frame of a clinical trial, and it's possible to get it in some specific places, like in France with the expanded program, early access program. Sorry. I don't see other, how can I say, reliably supported, scientifically supported approaches that we can pursue right now. So unfortunately, I would try idibenone. If idibenone does not work, I just would hope that something happens. I have seen cases which very slowly go down, they stabilize, and at some point also recover a little bit. So it's a timing issue. However, as Magali pointed out, we don't have any mean right now to predict how the prognosis will be in each single patient. The data presented are a summary of all the patients. So obviously you have, as for any drug, responders and non-responders. Allora, in italiano, a parte l'idebenone, che è la terapia approvata in Italia, che chiaramente può e deve essere testata, la terapia genica è imprevedibile poter dire chi risponde positivamente e chi invece ha un'esperienza negativa di non risposta. In questo momento non siamo in grado di predire questo né di come andrà. Tuttavia ci sono stati casi che hanno deteriorato lentamente e poi dopo un periodo di stabilizzazione hanno recuperato, quindi c'è anche una questione di tempistica che potrebbe essere differente in questo caso rispetto agli altri, e io non perderei tutte le speranze. E altre soluzioni terapeutiche che siano sufficientemente supportate dai dati clinici, preclinici, scientifici, che ci autorizzino a fare qualche cosa di più, non ci sono in questo momento, almeno non in mia conoscenza. Ci sono alcune sperimentazioni, per esempio le cellule mesenchimali, ma non c'è nessun dato a supporto chiaro e scientificamente validato a supporto. Esatto, a supporto di questa pratica, e io non la consiglio fino a che non abbiamo le idee chiare dal punto di vista scientifico. Gli spari nel buio non li consiglio mai in nessun caso. Ok.
Thank you for that, Dr. Crowley. I appreciate that. Well, so we're going to take two more questions and then we're going to close out. So the next question is, could a patient who has already taken part in gene therapy trial with no positive results take place in another gene therapy trial, for example, the Neurop one?
The short answer is no, because the gene therapy can be done just once. So if the approach is the same, it's not possible to do again this.
No, not only that, but
But it's-
it needs to be the same. The approach needs to be for the same mutation. You cannot jump from one mutation to the other.
Okay, thank you. Thank you for clarifying that. The last question is there a chance for someone who's over 18 months from the onset of disease to receive the inspection with Lumovoq?
Maybe Magali can answer to this point because it's kind of gray area.
Yes. So I think today, when we looked at the data, there is a space to have a treatment a bit later. I would say within 2 years. This could be done potentially through early access programs if the regulators are in agreement, because in our early access program, what we observed is that patients were treated later comparing to clinical trials, and the results are quite good even for patients treated later. In the future, it's not yet there, but our plan is to develop in chronic patients, meaning patients beyond 2 years post-vision loss, but it's not yet in the plan. But of course, it's something we would like to do later on, to develop in chronic patients with a visual loss, which is more 3, 4, 5 years ago. But it's not yet in the plan. We do need firstly, step by step, to get into this new trial, to move forward to approval with this therapeutic window. But of course, we need also to think and to take care of chronic patients.
Thank you for that. So that wraps up our program for today. I just want to say thank you to all of our speakers for the time that you've committed to this program today, and to the tireless work that you do to advance therapies and treatments for this community. We are truly, truly grateful for each and every one of you, and for your support of this community. So thank you so much for being here with us on this LHON Awareness Day. Paula, I'm going to turn it over to you to close us out.
Thank you, Kyra. I really want to thank everybody. I want to thank all of the wonderful people who dedicate their life to studying LHON, and I wish you wonderful work ahead of time. Hopefully we will come to a cure soon. I want to thank my wonderful board in IMP, and I would like to take this opportunity to invite you all to the 14th edition of the MitoCon International Conference, which this year is going to take place in Padua from Friday the 25th of October, that is October 25, to Sunday, October 27. You're all welcome to come. Just go on the MitoCon website, www.mitocon.it. And thanks again, IMP, for hosting this wonderful event.